Hi, hello, welcome to our second webinar on the series of SBBC and ISSCR. Before we start, I would like to thank again the people that made these webinars possible. First, Lígia Pereira from the Institute of Biology at USP. Lígia is a member of the ISSCR Board of Directors, and she introduced our idea to the ISSCR. We also want to thank Nancy Weedy. Oh, yes. So, sorry. I have to project my slides. I'm sorry. I will start again. Okay, so I'm restarting. So I thank Lígia Pereira from the Institute of Biology that made this possible. She introduced our idea to the ISSCR board. I was also thanking Nancy Weedy and Jack Mosher that embraced our idea and were of great support. And I also want to thank Keith Al, who recorded a video with some information about ISSCR. So you can watch the video at the SBBC's playlist on YouTube. And I also want to thank the speakers of this series that accepted our invitation. And the SBBC Board of Directors, especially Patricia Gama, our former president, and Irene Yan for all the help in putting these webinars and broadcasts together. And now I'd like to make two announcements. The first one is from ISSCR. They are inviting us to join members of the task force that were charged with updating the ISSCR guidelines for a 4-part digital meeting series. And so there are going to be 4 meetings, and so to discuss the new guidelines. And you have the ISSCR website that you can check and register for these events that are going to happen on November 10, 11, 18, and 19. And I also want to remind you that the deadline for abstract submission for our Congress, that's going to be in January 2021, so the SBBC Congress, is going to end on October 31st. So if you want to submit an abstract, you please do it, and you can access the Congress website on our webpage. And now I want to introduce our speaker for today, Professor Fiona Doestch, who I thank very much for being here with us today. It's an honor to have you. Dr. Fiona Doestch is a professor of molecular stem cell biology at the Biozentrum, University of Basel. And her research aims to decode the diversity of stem cell populations in the adult brain and to elucidate how physiological conditions modulate the maturation and behavior of neurostem cells. She is an internationally renowned expert in the field of neurostem cells, and she's well known by her work on neurogenic niche at the subventricular zone of the adult mammalian brain. She studied at McGill University in Montreal, Canada, and got her PhD in neurobiology from the Rockefeller University in New York. Prior to her appointment to the Biozentrum, she was a junior fellow at the Harvard University Society of Fellows and an assistant professor and then an associate professor at Columbia University in the US. Recently, in July 2020, she was elected as a new member of the European Molecular Biology Organization, EMBO. So, Professor Doestch, thank you again for accepting our invitation. It's a pleasure and an honor to have you here.
Thank you so much for this kind invitation, and I'm really delighted to participate in this seminar series. And I thank also Lígia for putting this together with the Brazilian Society for Cell Biology. So I'm going to try and share my screen and do this again.
Yep.
One moment. Come on. Okay. And do you see the correct mode?
Yes, everything's fine. Thank you.
Perfect. Okay, thank you. So I'm really delighted to share our ongoing research with you on the topic of stem cells in the adult brain. So as an introduction, you know that stem cells are found in many different organs in the adult body, and these are essential for maintaining tissue homeostasis, but also for repair after injury. And these stem cells live in very complex and specialized microenvironments or niches that support these cells, their function and behavior throughout life. So when a stem cell becomes activated to divide, it will give rise to transit amplifying cells, which are very rapidly dividing cells, and this increases the number of cells you get from a single stem cell. And these transit amplifying cells, in turn, then give rise to more differentiated progeny. So the niches that the stem cells inhabit are highly complex. They contain vascular cells and many other cell types. And an emerging theme that's come from the study of different organs is this concept that there are different pools of stem cells present, some of which are quiescent or deeply dormant, and some of which are activated or actively cycling. However, very little is known about what signals recruit quiescent stem cells to become activated, and also the lineage relationships between these different types of stem cells. So we're very interested in these questions in the context of the adult brain. And so I'll tell you about our work on adult neural stem cells. So in the brain, you have two major classes of cells: neurons, which are the cells that allow us to communicate and to move and to think, and then a much less studied cell type, namely glia. So glia are essential support cells in the brain, and there are two main types: astrocytes and oligodendrocytes. Astrocytes are star-shaped cells, and they get their names from this morphology, and they have many diverse roles, including formation of the blood-brain barrier, which stops signals from the blood getting into the brain, supporting the neurons by absorbing extra sort of signals around them, and supporting synaptic function. Oligodendrocytes are the cells that form the myelin sheaths along the axons of neurons, and this allows neurons to rapidly propagate their action potentials. So almost all neurons and glia are born during embryonic development. However, glia can continue to be made in the adult, and in particular, oligodendrocytes. And these oligodendrocytes are frequently thought to arise from oligodendrocyte progenitors, which are these highly branched or ramified cells found scattered throughout the entire brain. And they express two canonical markers: platelet-derived growth factor receptor alpha and NG2. So these cells can divide and give rise to new oligodendrocytes throughout life. So we now know, and it's been known actually for quite a long time, that new neurons continue to be added to certain parts of the adult brain. And this is really a widely conserved feature across almost all animals. But what's particularly interesting is that the levels of neurogenesis and the sites of neurogenesis are different between species. So in songbirds and in lizards and fish, you have fairly widespread neurogenesis, but in mammals, neurogenesis in the adult is much more restricted. And although stem cells are found in the human brain, it's still quite controversial as to the levels of neurogenesis that continue in the adult and the human. So all of the stem cells in these different organisms share a common identity. And we're going to talk today about the adult mouse brain, and in particular, a region called the ventricular subventricular zone, or VSVZ. So shown here on the left, you have a top-down view of an adult mouse brain, with the front of the brain, the olfactory bulb here at the top, and the back of the brain at the bottom. And what I've outlined here in dark blue are the lateral ventricles. And this is where the ventricular subventricular zone is located, immediately adjacent to the ventricles. Shown below, you can actually see here in light blue the lateral ventricles, and the cerebral spinal fluid fills the ventricles and flows through the ventricular system. So So you can appreciate from this perspective that in this top view of the brain, that the ventricle is a very curved structure. And so a very nice way to visualize the entire ventricle at once is to do a whole mount dissection, and then to stain these whole mounts. And when you do this, you find that the entire SVZ contains a beautiful network of chains of migrating neurons. So each one of these round circles is a newly generated neuron, and they migrate all the way forward through most of the length of the brain to reach the olfactory bulb, where they turn into different types of olfactory bulb interneurons. So this is a very robust process. About 30,000 to 100,000 new neurons are born every single day. And the SVZ also makes some glia, but much lower numbers. So if we take a cross-section through the mouse brain, this is what it looks like. And again, in the center, the white area are the brain ventricles. And then the SVZ is found immediately adjacent to it. There are two walls, the lateral wall and the septal wall. And the lateral wall is the most active, so you have many stem cells and a lot of cell division happening here. And this is the main site of neurogenesis. So if we zoom in on the cell types, we can see them here on the schema on the right. Lining the ventricles are ependymal cells, and these cells have long cilia that propel the cerebral spinal fluid through the brain ventricles. Then we have the stem cells, which have a radial morphology. They're shown here in blue. And we showed many years ago that the stem cells are actually a type of astrocyte. They express GFAP, and they have many other features of astrocytes. These we now know are found in quiescent and activated states, and upon activation, they divide to give rise to transit amplifying cells that then give rise to neuroblasts that migrate to the olfactory bulb. So this raises the really fascinating question of whether all glia, or all astrocytes or stem cells, or whether this potential is restricted to those that are found in stem cell niches. And likewise, what are the signals in the microenvironment or the niche that support ongoing neurogenesis throughout life? So these are questions we're very interested in the lab. And a key step to begin to answer these is that it's important to be able to actually purify the stem cells from the brain to test their functional and molecular properties. And so we put in a lot of time and effort to try and identify markers that would allow us to directly isolate the stem cells from the niche to be able to investigate them. And what we'd previously found was that epidermal growth factor receptor, or EGF receptor, marked the activated stem cells as well as the transit amplifying cells. But we didn't have a marker that allowed us to distinguish between the quiescent stem cells and the more typical niche or branchy astrocytes. And we found that CD133, or prominent, nicely labeled both the quiescent stem cells and the activated stem cells, but not the niche astrocytes. And shown here on the left, you can see one of these radial astrocytes that's been electroporated with a prominent promoter. So by combining these different markers, we were able to fact sort for the first time quiescent stem cells and activated stem cells and directly compare their properties. And we found that about 16% of the astrocytes were activated neural stem cells, and surprisingly, the quiescent neural stem cells represented a very large fraction, about 35% of the astrocytes. When we tested their in vitro stem cell properties, to our surprise, we found that the quiescent neural stem cells could not generate neurospheres very well, in contrast to the activated neural stem cells. And likewise, they didn't express nestin, but they then upregulated nestin as they became activated. So this revealed that these two canonical features of stem cells that are widely used in the neural stem cell field to identify stem cells, namely neurosphere or colony formation or nestin expression, were not detecting the quiescent stem cells. So we then compared the transcriptomes of the quiescent stem cells and activated stem cells, and this revealed this really interesting observation that the quiescent stem cell state is actively maintained. So the quiescent stem cells were highly enriched in cell-cell interactions, extracellular matrix and adhesion to the niche, G-protein-coupled receptor signaling, and they were very enriched in lipid metabolic pathways. So the quiescent stem cells are actively sensing the environment and integrating these signals to decide whether to maintain the quiescent state. So through our studies on these purified stem cells, we've come to realize how important stem cell heterogeneity is. And there are different types of heterogeneity. The first is the morphology of the stem cells is quite distinct. So shown here are 4 different pictures of different shapes of stem cells, and this highlights that these cells will interact with different compartments of the niche. We then also know about the proliferation state. So we have the quiescent and activated stem cells. But what's still very unclear is whether under homeostasis and regeneration, it's the same pool of stem cells that's active, or whether during regeneration or injury, a separate reserve pool of quiescent stem cells is turned on to then regenerate the lineage. Finally, another key aspect of heterogeneity within this niche is the regional identity of the adult neural stem cells. So shown here in different colors are different spatial domains that give rise to different types of neurons. So depending on where the stem cell is located, it will give rise to either a deep granule neuron or a superficial granule neuron or different subtypes of periglomerular neurons. What's not clear is whether similar domains also exist for gliogenesis. So in the lab, we are tackling these questions of stem cell quiescence and activation, and what are the balance of the intrinsic signals and the niche signals that regulate stem cell behavior. I'd like to just briefly tell you about some interesting findings we have on the stem cell niche, which have begun to shed new light into the regulation of adult neural stem cells. So the SVZ lies between 2 layers in a whole map perspective. First, you have the ependymal cells, which line the surface of the ventricles, and they're multiciliated. So that here you can see these lots of dots. These correspond to the basal bodies of their cilia. And in beta-catenin, you can see the outline of the ependymal cells. And the ependymal cells form these beautiful pinwheel structures over the wall of the ventricle. And the stem cells actually directly contact the ventricle and the CSF at the center of these pinwheels. So you can see the stem cells here with a single puncta. The stem cells then here are shown in the middle, and they have an apical process that touches the ventricle, and then a basal process that touches blood vessels. And shown here on the right is a specialized planar vascular plexus that's found in the SVZ that extends across the whole wall. And you can see in green, the dividing cells are very tightly associated with these blood vessel structures. So in between the ependymal cells and the vasculature, you have all of the lineage cells. So the transit amplifying cells, the neuroblasts. You also have other cell types such as microglia, many extracellular matrix structures. And this just highlights how important the local cell-cell interactions are between the lineage cells. However, we've begun to understand that long-range regulation, so signals from outside of the niche, are very important in regulating neural stem cells as well. So first, we found that the stem cells have a specialized vascular niche. So unlike the rest of the brain, where the astrocyte end feet surround the blood vessels, we find that the stem cells and transit amplifying cells actually directly contact the blood vessels at sites that lack the astrocyte end feet. So they are mediating or integrating signals that are contact mediated and diffusible signals. And furthermore, factors in the blood are actually able to access this part of the brain, which they can't do in most areas of the brain. The stem cells also send a tiny process to touch the cerebral spinal fluid, and they have a primary cilium that can detect factors in the CSF. So shown here, you have the the ventricles, and the CSF flows through the ventricular system. And the CSF is made by a structure floating in each ventricle called the choroid plexus. And this is a highly vascularized tissue that really forms a barrier between the blood and the CSF. And what we found is that the choroid plexus is actually also a key source of secreted factors that are secreted into the CSF and then regulate all stages of the lineage. And very interestingly, the secretome of the choroid plexus actually changes in different states. For example, with aging, we found that the secretome of the choroid plexus changes, and that age factors inhibit the division of young stem cells, whereas young factors can actually stimulate the division of old stem cells. So this highlights how important the choroid plexus is in regulating neural stem cells. And we actually believe that it may be integrating signals from the blood and also the brain to then dynamically change its secretome to then regulate the stem cells. So finally, another insight into long-range signals came from a small molecule screen we performed to identify activators of quiescent neural stem cells. And the main class of compounds we found that stimulated neural stem cells were actually neurotransmitters. And this led us to this quite intriguing finding that actually neurons in other parts of the brain can actually send long-range projections specifically to the stem cell and niche and regulate the division of one pool of stem cells. So for example, here we found that the hypothalamus POMC neurons, which are important for feeding behavior, send projections to regulate the division of NKX 2.1 positive neural stem cells. And this leads to the specific generation of one type of deep granule neurons. Very interestingly, just hunger and satiety itself also had the same effect. So this could regulate the division of one pool of cells. So this really suggested this idea that these long-range neural circuits could be coming in and mosaically innervating the SVZ and regulating different pools of stem cells. And leads a new dimension to our understanding of the niche, namely that physiological state itself can also regulate the division of specific pools of stem cells. So now I'd like to tell you about a new study that we're putting together about a new regulator of adult neural stem cell quiescence, namely platelet-derived growth factor receptor beta. So we had found this receptor by comparing the quiescent and activated stem cells, and it was very enriched in the quiescent stem cells. Now, PDGFR beta is well known to be expressed in parasites throughout the body. And so we were quite surprised to see that it was so enriched in the SVZ quiescent cells. So first, we confirmed that it's expressed in the SVZ. And you can see here on the right, the green staining labels very brightly the SVZ, as well as parasites throughout the brain. So we then performed co-staining, co-immunostaining, and found that indeed PDGFR beta, shown here in the left on green, is expressed in the stem cells at the center of the pinwheels. And on the right, you can see that they have the typical radial morphology of the stem cells when you express mCherry under the PDGFR beta promoter. So we then checked which other stages of the lineage express PDGFR beta and found by co-staining for EGF receptor, and double cortin, which labels the newly generated neurons, and MCM2, a cell cycle marker, that PDGFR beta was really restricted to the quiescent stem cells and some activated neural stem cells. So we found that the transit amplifying cells here on the left in red did not express PDGFR beta. The neuroblasts in this middle panel in red do not express PDGFR beta. And likewise, the MCM2 cells on the right in red are also not expressing PDGFR beta. So this reveals that PDGFR beta within the lineage is expressed predominantly by the quiescent neural stem cells. So we next perform fate mapping using lineage tracing by using PDGFR beta Cree ERT2 mice. So we can selectively induce tracing of the cells in the adult by injecting tamoxifen, and then we analyzed the brains 1 month or 4 months later. And we looked in the SVZ and in the olfactory bulb and the corpus callosum. So neurons go to the olfactory bulb, and the corpus callosum is the site of oligogenesis, or the destination of oligos. So here you can see beautiful radial stem cells labeled, and 1 and 4 months later, you have new neurons in the olfactory bulb and also new oligodendrocytes in the corpus callosum. So these cells give rise to new neurons and oligodendrocytes with slow kinetics. So we next wanted to know, what is the function of PDGFR-beta in regulating neural stem cells? And so to selectively delete PDGFR-beta in neural stem cells, we used GFAP Cre-ERT2 mice, which upon the addition or injection of tamoxifen lead to deletion of the ligand binding domain of PDGFR-beta and the expression of tomato, a fluorescent protein that allows you to trace the fate of the cells. So when we gave tamoxifen and looked 1 day later, we found that actually there was a great increase in the number of dividing stem cells. And this was accompanied by an increase in the number of activated neural stem cells and transit amplifying cells. So by deleting PDGFR-beta, you actually release the stem cells from quiescence, and they become activated to divide. As a consequence of this, if you look 45 days or 6 months later, we saw more new neurons in the olfactory bulb in the mutant mice compared to wild-type mice, and similarly more oligodendrocytes in the corpus callosum. So together, these results reveal that PDGFR-beta maintains the quiescent state of adult neural stem cells. Now this is quite interesting because we know that the septal wall is largely quiescent under baseline conditions. And so we wondered whether we could use these mice as a new tool to identify domains that are normally quiescent under homeostasis. And indeed, it turned out to be the case. So just to expand this, I need to remind you of the different domains that have been identified so far. So here we have a brain section. The SVZ is next to the brain ventricles. And in green, you have the neurogenic site. So many olfactory bulb neurons are born along the lateral wall. And in the dorsolateral corner in orange, you have a site of oligodendrogenesis. However, it's not known whether astrocytes are generated in the adult SVZ. And by analyzing our mutant mice, we actually uncovered several novel gliogenic domains, both for astrocytes and for oligodendrocytes. So first, when we looked at astrocytes, we found a domain uniquely on the septal wall with a new cell type, which we call gorditas, because they have a small rounded soma and sort of plump morphology, and they have bright GFAP positive processes. And in the mutant mice, these were really selectively increased in the septal domain. And if we looked 45 days later, we saw a great increase in the number of astrocytes, specifically in the septum, not in other brain areas. Next, we looked at oligodendrogenic domains. And as I told you before, the dorsolateral corner is well known. And in our mutant mice, we found that actually the entire septal wall, but especially this dorsal septal corner, was very enriched in oligogenesis. And most intriguingly, we identified a new domain that was actually inside the brain ventricles, so intraventricular. So you can see here with the arrows, tomato-positive cells that co-express the marker OLIG2. So all of these domains were enriched in the P-beta mutant mice. So I'm going to tell you a little bit more about this intraventricular oligodendrocyte progenitors, because they were a very intriguing new cell type. So where do they come from? in our mutant mice, we found that they were often present near these large clusters of GFAP-positive, tomato-positive cells with OLIG2 cells near to them. We wanted to know whether they also existed in wild-type mice and were not simply a result of them deleting PDGFR-beta. And indeed, in wild-type mice, in cross-sections, we could again see these small clusters of GFAP-positive cells in red, and now using a different marker, NG2, to mark the oligodendrocyte progenitors, we could see them tightly associated with these GFAP-positive cells. But these are rather difficult to see in cross-sections. So we turn to our whole map preparations. And here you could beautifully see, on top of the ependymal cells, which are labeled in blue, these intraventricular oligodendrocyte progenitors in green. Here they're stained with the marker PDGF receptor alpha. When we did staining for ciliary markers, we could see again that they were nestled among the ependymal cell cilia, sometimes as individual cells, and sometimes found as little clusters. And when we did co-staining for GFAP and PDGF receptor alpha, we often found these cells associated with the large clusters of GFAP in red and PDGF receptor alpha in green, sometimes with a single radial cell and sometimes more dispersed. Now, interestingly, they were largely located in the more anterior or rostral portion of the ventricle, so they're not found all over the wall of the ventricle. And finally, when we compared them to other oligodendrocyte progenitors elsewhere in the brain, it was clear that they have a different morphology. So they're much larger and smoother than the more typical ramified oligodendrocyte progenitors in the striatum or even a bit deeper in the SVZ. So we think that they're a really intriguing novel type of oligodendrocyte progenitor. So what might they be doing? It's recently been described that there are supraependymal axons. So this means axons that are actually on top of the ependymal layer that come from other brain regions, including serotonergic axons from a part of the brain called the dorsal raphe nucleus. And so we asked whether these intraventricular oligodendrocyte progenitors might be interacting with these axons. So we performed immunostaining, and here you can see that these OPCs in red are clearly wrapping around and touching the green axons. And many of these were immunostained for serotonin, shown here in red. Finally, we didn't see any myelinating fibers on the wall of the ventricle. Here in the green labeling is a nicked bit of tissue where you can see lots of labeled myelinated axons. So these really suggest that this intraventricular oligodendrocyte progenitor might be interacting with the axons of these cells, and they're uniquely poised to send signals in the CSF. So we're very interested now in these new domains, and particularly oligogenic and astrogenic domains, and would like to understand how they might be contributing to glial plasticity and perhaps adaptive gliogenesis in the brain. So finally, I'd just like to tell you a little bit about some of our ongoing work about understanding now neural stem cell heterogeneity at the molecular level. And we were very interested in defining more about markers for the different cell types in the lateral wall and septal wall, but in particular also whether there might be differences between males and females. And so we simply dissected the lateral wall and septal wall from male and female mice and performed large-scale single-cell RNA sequencing of unsorted cells. So this captured all of the lineage cells and all of the niche cells. And it provides a very nice resource to investigate the different cell types and their heterogeneity. And this revealed multiple types of glia, multiple neuronal and oligodendrocyte lineages. But very intriguingly, it also suggested that males and females may have different signals that regulate their stem cells, including neurotransmitter receptors and GPCR-coupled receptor signaling. So summarizing this large-scale data set, we again found that the septal wall is more quiescent and gliogenic, whereas the lateral wall is more proliferative and neurogenic. Interestingly, there are more oligodendrocyte progenitors in males, and that there's still a large amount of intra-regional heterogeneity that we have to uncover. But together, this really highlights how important it is to actually consider both males and females in studies. So in the future, we'd like to understand what signals cause the on-demand recruitment of stem cell pools in the adult. We found that one domain in the SVZ responds to hunger and satiety. And very recently, we found new domains for oligodendrocytes, including these intraventricular oligodendrocyte progenitors and astrocytes. And we'd like to understand, are there other states in males or females that may recruit different pools of stem cells? And what are the niche signals? Could they come from the blood vessels, neural circuits, or the choroid plexus? And are there differences between males and females that might potentially lead to the recruitment of different pools of cells for specific behavioral functions? So finally, I would like to thank my wonderful colleagues in the lab, first at Columbia and now at the Biozentrum, and also the many sponsors of our research over the years. I'm very happy to take any questions. Thank you.
It was wonderful talk. Beautiful, beautiful images. Your photos they're amazing.
Thank you.
We have several questions.
So I'm going to exit so I can see you. So if that's okay.
Yes.
Because otherwise I can't see anything.
Yeah.
Okay, stop screen sharing.
Yeah.
Does that work? Yes. Okay, great.
Okay, great. So thank you so much.
Thank you.
And so I'm going to start. Okay, so the first question was from Julio Roncatov. He's asking, so with this large amount of distribution, why they don't help the against multiple sclerosis?
Yes, so there are a large number of stem cells, but many of them seem to be committed to neurogenesis. So we used to think that all stem cells were multipotent and they could give rise to neurons or oligodendrocytes or astrocytes. But I think the idea that's emerging now is that they're more restricted than we used to think. But clearly there are some that are recruited. So if you do a demyelinating lesion, for example, in the mouse, you do have stimulation of many oligodendrocyte progenitors, including the SVZ stem cells that go up and remyelinate in the corpus callosum. There are, of course, other factors with demyelination that then, block it. So the hope is that one could eventually understand more about recruiting these.
Thank you. The next question by Irene. How important is the BBB, for instance, in this permeability in modulating the SVZ niche?
Yeah, so this is a really fascinating question, which we haven't pursued far enough yet. So what we found is that factors in the circulation can enter the SVZ, but we didn't actually define which factors. So we injected different sizes of tracers and found that the larger the molecules, fewer appeared to get in. But very recently, there was an interesting study suggesting that actually even in young mice, there's large-scale transfer of serum-derived factors into the brain, which people had just not appreciated. So I think it would be really interesting now to go back and explore more carefully by tagging all of these different factors to see what gets into the brain. I do think that it's important in regulating them.
Yeah. Next question by Claudia Batista. She's asking about expression of PDGF or beta in the dentate gyrus.
Yes, so it's also expressed there. We haven't done the detailed analysis we've done in the SVZ, but with lineage tracing, we definitely saw the radial astrocytes in the hippocampus, and also further back, so in around the third ventricle, for example. So we think that it may be a common regulator of more quiescent pools within the brain.
And the next question was by Claudia as well. If you have any data on cannabinoids regulating SVZ neurogenesis?
No, we have not looked at that. Yes, okay. Interesting question.
Yeah. So the next one, could male versus female differences be an embryologically originated difference or differences in response due to hormonal differences?
Yes, absolutely. So one thing I didn't mention is that these adult stem cells actually enter quiescence very early, so around embryonic day 13. So clearly there are patterns being set up in the embryo. So how much of that is already specified early? How much changes with puberty, for example? We don't know at the moment, but it's a really interesting question. And we're actually trying to directly test male and female cells and male and female niche signals in the adult to understand some differences that may exist, at least in the adult. But how early they're set up is still open.
Okay. Thank you. Now, another question by João Menezes. What about psychedelic serotonin activation molecules such as LSD? Would they increase intraventricular oligodendrocyte precursors?
Yeah, we're fascinated by this interaction with the serotonergic fiber. So we don't know. This is all extremely new. But even to understand if, for example, you stimulate these fibers, what happens then to the OPCs? We haven't manipulated them in any way. But one could try and look at some of these compounds and see how they influence the cells in vivo.
Okay. So one more. I told you we have several.
That's great.
yeah, it was really, really interesting. So Silvana Lodge, she's saying that in crustaceans, one of the signals that seem to activate adult neurogenesis is serotonin. That you mentioned serotonergic axons, but do you think it may be one of the key molecules to trigger this event in mammalians?
Yeah, so in the paper I mentioned where they identified these serotonergic fibers along the wall of the ventricle, they actually showed that it did regulate the proliferation of adult mouse stem cells as well. So when we did our screen, we found that multiple neurotransmitters, actually different types, seem to activate different stem cells. And so whether it may regulate one pool of stem cells or all of them is still unclear. But I think that it definitely will be a key player among some of these stem cells.
Okay. Patricia Gama is asking, when you regulate BDGF or beta, have you studied the correlation with cell site control proteins that are related to quiescence, for instance, P2N1?
Yeah, so this is a very interesting question also. So what we have begun to do is FACS sort the cells from our wild type and our mutant mice. And we've actually done ATAC-seq as well to try and understand how the chromatin landscape might be changing. And what's quite interesting is that there seemed to be a distinction between the activation step and the cell proliferation steps. So you can really untangle these two events. So yes, in the mutant, eventually you do have fewer cell cycle inhibitors and more proliferative markers, as well as these interesting chromatin remodeling of the landscape that probably accompanies the activation from the quiescent state.
Okay. And now a question by Jack Motion. Hi, Jack.
Hi, Jack.
A very interesting talk. Is the behavioral state, hunger society, driving increased neurons through increasing TA cells or mobilizing the quiescent stem cells?
Yeah, so the way we came to this was doing a screen on the quiescent stem cells, and we clearly see that, so we looked in vivo, and acutely we saw an effect on the stem cells, but we also a bit later saw increased transit amplifying cell proliferation. So whether it's uniquely acting on the stem cells, we don't think so, but the receptor is highly enriched on the stem cells and also more ventrally. So we think it's a combination.
Okay. Now, one more question by Cecilia Edin. Are oligodendrocytes generated with activity regulation in the SDZ?
Yeah, this is a great question, and we don't know the answer yet, but absolutely. There's lots to study.
Yeah.
Yes.
So I have one question myself. Do you think that BDGF or beta is also playing a role in the differentiation of astrocytes in outside the SBZ.
Yeah, so I didn't mention that, but PDGFR beta is expressed in some astrocytes. In our mutant mice, because we use the GFAP CRE-ER, we're also affecting it there. And if we look, for example, in the cortex, we didn't see any change or any proliferation. But we did see in the septum some changes. So it could be that the septum is like a uniquely plastic state that's somehow regulated by PDGFR beta signaling. So it's possible that other astrocytes are affected. It would be very interesting to look in this model now, using this mouse at different injury models or lesions to see, do you actually enhance the response compared to a normal state? So these could be very interesting to look at in the future. Oh, I can't hear you.
I think Marimeli's connection went off at some point. Hold on a second. Oh, sorry. I'll just leave the room and see what happened.
Okay.
Oh, I'm back.
Hello.
Sorry, I just my internet crashed.
Yeah.
Yeah, so I'm not sure that now I miss it. The chat. I'm not sure if we have any more questions. Irene, you have your mic is off. Your microphone is off. Irene. Yeah.
No, we're okay. Most of the when you left the room, the conversation was around. Oh no, Marimeli disappeared. So we're good.
Oh, okay. Do we have any more questions? No, Patricia. Oh, Patricia is saying that we don't have any more questions. Yes. So Fiona, thank you so much. It was really nice finally meeting you. You know, I know a lot about your work. All my students have read your work, and so it was a great pleasure to have you here with us.
And for me too. I really enjoyed it, and I look forward to more interactions in the future as well.
Yeah, we do too. Yes. We hope that someday we can bring you here in person.
I would love that.
Yeah. Okay.
Yeah. Great.
So thank you so much
again,
and thank you, Jack. You know, from the ISSCR for making this contact and making this possible.
Thanks.
Okay. Bye.
Bye-bye.
Thank you.